Haider bin Sharaf al-Musawi, an Omani PhD student in the field of molecular pharmacology at the University of Strathclyde, was able to provide a modern method for treating brain cancer in his current study in the UK, Glasgow. Modern method is based on the selective inhibition of neurological signals inside the cancer cell, adding that he is currently diagnosing and treating cases with a genetic mutations that is the highest in patients with this type of cancer. Haider bin Sharaf al-Musawi, who is gifting his achievements to Oman and His Majesty the Sultan, is a graduate of bachelor's degree in pharmacy from the University of Strathclyde in 2011. He obtained a clinical pharmacy master's degree from Robert Gordon University in 2016 and works as a lecturer and researcher in the pharmacy program at the Oman College of Health and Science. And now, live with us on telephone call is Haider in Glasgow. Haider, good evening to you.
Hello and good evening. Thank you for having me this evening.
Thank you very much. And first of all, we'd like to say congratulations for your achievement in your study. And tell us, first of all, how are you keeping up with the world pandemic? Are you okay there?
We are coping well with the current situation and sticking to the rules and restrictions applied by the UK government to handle these circumstances that we all hope pass soon.
So you are actually following the precautionary measures which are kept there in the United Kingdom?
Exactly, exactly.
Well, can you tell us about your study then? What is glioblastoma and why glioblastoma?
Well, glioblastoma is actually a grade 4 brain cancer and is considered one of the most aggressive types of cancer with a mean survival period of 15 months with the current therapy approach. Mm-hmm. And it's worth mentioning that the number of cases in Oman increased by about 20% from 1996 to the year 2015. So that's why I chose glioblastoma to try and come up with a novel strategy to help either treat the disease or prolong the survival with better quality of life.
Mm-hmm. And what is the nature of your study exactly?
Well, my study is actually divided into two phases. Mm-hmm. So phase 1 was to prove that there is a very specific cell signaling in the cancer cell, and it is active. Mm-hmm. And then we can use it as a potential target for new drug therapy. I managed to knock down this cell signaling pathway, which is called the NF-kappa B pathway. Specifically speaking, one part of it, which is the non-canonical pathway, using first-in-class, in-house made small molecules as a novel compounds to treat cancer. And the preliminary results are really, really promising. And the second phase comes now, while I'm examining the effect of such compounds and molecules on the phenotypic characteristics of cancer cells, such as cell growth, invasion, metastasis, and most importantly, immortality. And that is linked to the gene mutation HTERT, which is found to be found in more than 55% of the cancer cases. In this phase also,
okay. Continue.
So, in this phase, I will also diagnose specific HTERT mutant cells and then try to knock down these signaling pathways in such a subpopulation of patients using more sophisticated laboratory and clinical examination and tests.
Mm-hmm. Is this the only study that has been conducted, or there are other studies conducted similar to this?
It has been conducted on other cancer cell types like the pancreatic cancer, the breast cancer, and prostate cancer, but it is the first in glioblastoma.
Wonderful. Now, what is next then tell us next.
Well, next we'll be using the first-in-class, potentially novel anti-cancer small molecules to treat those subpopulation of patients with such compounds for a long-term therapy approach that is more specific to the H30 mutation. And it will also help to provide a new diagnostic tool in glioblastoma cell lines.
Well, Haider, thank you very much. Well done and great achievements. We wish you all the best and keep it up. Stay safe and healthy. Oman needs you. We are waiting for you to come back. And thank you once again.